You are walking on a plane and suddenly you encounter a wall that is infinitely high, infinitely deep, infinitely wide to the left, and infinitely wide to the right. What is this wall? This wall is death. This quote from The Wandering Earth vividly describes the powerlessness and despair humans feel when facing death. It is the unavoidable end of life. We often say that life should be meaningful, so that when we grow old, we won't have regrets. It seems that meaningful things are meant to provide a positive and peaceful recollection of one's life when old. However, aging is not an eternal state. What follows aging is death. Regret or satisfaction in old age, along with all your wisdom, knowledge, understanding of the world, and personal character and mindset, will all disappear. This is undoubtedly the most terrifying aspect of death. Myths and imaginations can be romantic and full of humanity, but reality is always cold, and facing this reality requires courage. After crossing the wall on the plane, no matter how much people on this side of the wall shout, there will be no response from the other side. A grand funeral is for the living. It has no effect on the deceased. There is nothing we can do in the face of death. All religions interpret death as some form of eternal life, where the consciousness of the deceased continues to exist in some manner. This is the hope of the living for the dead, and also their expectation for their own future fate. Eternal life is a human pursuit, and religion caters to this by explaining death as the beginning of the next phase rather than the end of everything. To alleviate the despair and loss of meaning people feel when facing death. However, the method of understanding the world through observation, logical reasoning, and research has now taken root. Humanity, while gaining great power, is also confronted with a harsh reality. Memory and consciousness are energy conversions at the cellular level. When the body dies, the heart stops beating, and blood no longer supplies oxygen to the brain. Mitochondria run out of materials to produce energy, and all cell functions cease due to lack of energy, resulting in the loss of any sense, thought, memory retrieval, or cognitive function in the brain. According to current scientific research on the human body and brain, there is no consciousness that can exist independently of the body. Humans are not spiritual beings. We are more like machines. When a machine made by ordinary humans breaks down, it can still be repaired because it is relatively simple. The parts of the human machine, however, are the size of a needle tip, with 40,000 such parts fitting into a small space. The human body is composed of approximately 60 trillion such parts working in coordination. Current human technology cannot perform precise repairs at such a small scale. Once the human machine experiences a systemic problem, and the cooperation between cells is forced to stop, billions of cells can fail simultaneously in a chain reaction. Completely repairing this highly complex human machine is almost impossible. So death for a conscious individual is the end of everything. The average human lifespan began to increase after the Industrial Revolution. In the 1950s and 60s, following World War II, world order was established, and there were no more wars, ushering humanity into a golden age. The average lifespan increased rapidly, from 44 years in the early 1960s to 77 years today, a gain of over 30 years in 60 years. Now, those of us who are teenagers or in our 20s might wonder how much our lifespan will increase in another 60 years. With more time ahead of us than our predecessors and advancements in medical technology, our lifespan could continue to increase, possibly even achieving the so-called eternal life. But is this possible? Since the 1960s, the overall rate of lifespan increase has been declining. While lifespans are still increasing, they no longer show the same strong trend as before. There is another curve related to lifespan that has not been displayed, the curve of maximum human lifespan over different years. Our average lifespan has more than doubled in the last 150 years, But the maximum age of the oldest people has only increased from 111 years to an average of 117 years during this period. As early as 1899, there were records of people living to 110 years old. Further back, there were centenarians, but no records. The current Guinness World Record for lifespan is held by Jean Comment of France at 122 years, achieved in 1997, and has not been surpassed since. In other words, the increase in our average lifespan has not pushed up the maximum human lifespan. In other words, technological advancements and improved living standards have reduced the likelihood of dying from famine, violence, or plague, and have conquered more and more stubborn diseases, allowing humans to live to our natural lifespans. However, this does not grant us an unlimited lifespan. It seems that there is a limit to human lifespan that modern technology cannot break. Now, the question is to understand why this limit exists. Let's explore the limits of life. Can humans achieve eternal life? Currently, there is no unified understanding in the scientific community regarding the causes and mechanisms of aging. There are many different theories, but the truth about aging is gradually being revealed. People's understanding of aging and death is improving under the guidance of scientific inquiry. Yuval Noah Harari, in his predictions for the future, asserts that humans will soon gain the ability to live forever and suggests that there might already be individuals among us who will achieve this. Given the current level of scientific understanding, is it really possible in the future to avoid aging and natural death? Or perhaps, will death be redefined, with no more so-called natural deaths, and aging being treated as a malignant disease that can be cured? To investigate this issue, I believe we need to start from the emergence of life and see if the death of individual organisms is inevitable. Previously, my focus was more on how organisms adapt to their environment, primarily studying how they live, but I had not seriously considered aging from the perspective of life's evolutionary history. Do all organisms have a lifespan limit? For 3 billion years after the appearance of life, organisms existed in the form of single-celled organisms. It wasn't until 500 million years ago that multicellular organisms became more active. For early single-celled organisms, their main method of reproduction was binary fission, where they replicated their genetic material and then split into two. For a simple single-cell bacterium, in ideal environmental conditions, binary fission produces two identical cells. So, can we say that the original old cell no longer exists? Or has the life of the original cell been inherited by these two new cells? This depends on how we define it. One could say that the original old cell has died, or that its life has been perpetuated by its increasing number of descendants. Such organisms do not have a concept of lifespan because there is no individual death in this process. This is a form of immortality. For example, cyanobacteria reproduce in this way. They have existed on Earth for 2.6 billion years, producing oxygen, and continue to exist through binary fission. If we view this mode of reproduction as the perpetuation of life, we could say that a cyanobacterium from 2.6 billion years ago is still alive today. This simple and efficient method of binary fission shows no signs of natural aging or death. Why haven't modern organisms inherited this ancient, seemingly grief-free trait? Initially, primitive organisms were essentially immortal, but now so-called advanced life forms experience inevitable aging and death. Why must we die? To answer this, we need to investigate transitional organisms, i.e. microorganisms that exhibit individual aging and death. Are there such organisms? And if so, how do they age and die? After extensive research, many have been found. Firstly, Escherichia coli. Coli divide symmetrically, producing two daughter cells that are identical in size and shape. However, as these bacteria grow, they are subjected to environmental damage affecting their DNA, cytoplasm, and cell membranes. During division, This damage is not evenly distributed between the two daughter cells. All the damage, such as oxidized proteins, broken DNA, and damaged cell membranes, accumulates in one of the daughter cells. This cell, burdened with accumulated damage, grows and metabolizes more slowly, has a longer division cycle, and is more likely to die due to environmental factors, effectively aging. The other daughter cell is completely new and undamaged, essentially starting as a brand new E. coli. To obtain a completely healthy individual, one cell acts as a repository for damage and degradation. This is known as the garbage can theory. Each division results in one cell inheriting all the damage, allowing the other cell to remain completely healthy. From an anthropomorphic perspective, the cell inheriting the damage can be considered the parent, while the healthy cell is the offspring. The parent continually produces healthy offspring while accumulating all the damage until it eventually ages and dies, taking all the accumulated damage with it. Imagine if the division was completely symmetrical, then all the damage would be equally distributed to both offspring, without a mechanism to remove the damage. This would result in damage accumulating in the offspring, eventually leading to widespread apoptosis, or even the extinction of the entire population. The fundamental logic of evolution is not to protect the individual but to safeguard genetic information. As long as traits related to this logic support evolutionary survival and gene transmission, these traits will be amplified within the population, even if they include aging and death. To more effectively remove damage from the population, many eukaryotic organisms with cell nuclei, like yeast, have evolved budding reproduction. In this process, the budding offspring are much smaller than the parent cell, which acts as a garbage can mother cell, facilitating the retention of damage within the mother cell. An even more advanced example is the ciliate, which has two types of nuclei, one for metabolism and one for reproduction. During reproduction, the reproductive micronucleus is passed to the next generation, while the metabolic macronucleus is not replicated. The micronucleus carries the genetic material, forming the new cell's macronucleus, while the old macronucleus, representing the discarded somatic cell, remains in the mother cell. This further ensures that most DNA damage stays in the mother cell, which eventually ages and dies, consistent with the garbage can theory. By observing transitional organisms between immortal single-celled organisms and our lifespan-limited multicellular organisms, we see that even if an organism itself does not age and can be immortal, environmental limitations are unavoidable, and life is constantly subjected to environmental damage. If an organism is immortal through binary fission, environmental damage will accumulate over time within the entire population. You may not die, but environmental limitations will eventually kill you, and this accumulated damage will affect the entire population, leading to its eventual extinction. Individual aging can absorb damage and draw environmental hatred, preventing the entire population from being wiped out. Natural selection will favor evolutionary directions that ensure stable genetic replication. Imagine two types of organisms. One inherits diseases at birth, while the other gains new vitality. Which one is more competitive? This explains why immortal single-celled organisms are at the bottom of the food chain, with their overall numbers controlled by numerous predators. The theory that the parent retains all the damage to protect the offspring seems to echo the root of parental love. However, as organisms evolved into larger multicellular forms, the situation became more complex than the garbage can theory suggests. In multicellular organisms, only germ cells are responsible for division and reproduction through meiosis, while other cells perform their functions to protect the reproductive process. Compared to single-celled organisms that divide every few minutes, multicellular organisms reproduce much more slowly. Before reaching reproductive age, cells need to stay healthy, but environmental damage does not favor slow reproduction. Thus, Genetic mutations and natural selection have driven the evolution of another direction: cellular self-repair. The self-repair mechanism in cells starts working at full capacity after we are born, replacing damaged proteins, repairing UV-damaged DNA, and mending broken cell membranes until we pass the reproductive age, around 45 years old. After the so-called reproductive age, the task of cellular self-repair is considered complete, and the body no longer operates at full capacity. At this point, the aging damage garbage can theory takes over, removing damaged individuals from the population. Essentially, this removes cells with compromised repair abilities, combining diminished repair capacity with the garbage can theory to form the reasons for our aging and death. But since somatic cells can self-repair, can they keep self-repairing indefinitely, preventing aging and death, allowing us to live forever? To answer this question, we need to examine how this repair mechanism works. As we mentioned earlier, in binary fission, single-celled organisms try to leave all the garbage and damage in one of the two resulting cells. This damaged cell can be seen as the parent, accumulating damage over time until it eventually dies, taking the damage with it. If the damage were evenly distributed between the two cells, it would continuously accumulate within the population, leading to widespread death. However, some astute observers have proposed that the garbage can theory could be interpreted as the old cell creating a replica to take away the garbage. How do we distinguish between new and old cells? When cells divide, a new cell membrane forms in the middle. Experiments have shown that during subsequent divisions, the damage migrates toward the side with the older membrane, while the cell on the side with the new membrane is completely new. This seems to distinguish between new and old cells. Hence, the appearance of aging and death in the parent cell is to remove the accumulated garbage from the population. You might say that this applies only to single-celled organisms and can at most be analogous to current microorganisms. But can this theory be traced back to multicellular organisms like plants and animals? Multicellular organisms evolved from single-celled ones, and every animal and plant, including humans, develops from a single cell. During the production and development of somatic cells, billions of cells work together to ensure that the germ cells can develop healthily, allowing the next generation to be born. Could this be seen as an extension of the garbage can theory, suggesting that humans' consciousness and unconditional love for offspring, even to the point of self-sacrifice, stem from this ancient division phenomenon? The fundamental logic is that it is difficult to maintain DNA on a material level due to inevitable environmental damage. However, preserving the genetic information within the DNA is simpler. Creating new carriers to inherit the genetic information, with each carrier living and dying, allows the information to be relatively stably preserved. Saying that genes manipulate life is somewhat anthropomorphic. DNA is just a large molecule with no consciousness or volition, but under the right conditions, DNA can create conscious life. As long as the genetic information in DNA can be preserved, life will continue to exist. Gene mutations, or information replication errors, create diverse life forms. These different life forms are subject to environmental selection through survival of the fittest, leading to evolution. It is not the individuals best suited for survival that are selected by nature, but those best able to inherit, preserve, and transmit genetic information. This is a survival algorithm, also known as an evolutionary algorithm, a completely natural process. Many people cannot understand how such a simple principle can create such complex beings like us. This is because they also cannot grasp the concept of time: 3.5 billion years. Even the slowest chemical reactions and the smallest probabilities can achieve their outcomes over such a vast time span. However, the ancient survival principle of preserving a string of information remains unchanged. All individuals must reproduce new information carriers while absorbing environmental damage, and dying to prevent the next generation from being harmed by accumulated damage. Our cellular repair mechanisms aim to extend individual lifespan, but are also based on the principle of protecting the next generation. Self-repair in cells ensures individuals grow strong enough to better support the next generation. Maintaining old individuals' functions requires consuming more energy, so repair mechanisms work at full capacity until the reproductive age, after which they no longer function fully to save energy. Natural resources are meant to sustain an unbroken chain of information, not individual immortality. Organisms that do not adhere to this principle have gone extinct. A long-living individual might consume many resources, and if it dies accidentally, it increases the species extinction risk. Various animals have different lifespans. For example, among mammals, some mice live only up to 3 years, while others, like us, can live up to 70 to 80 years, and some, like the bullhead whale, can live over 200 years. These organisms seem to have longer lifespans as they get larger, which is generally true but not entirely accurate. Larger animals usually have longer developmental periods until sexual maturity, so natural selection has adjusted their lifespans accordingly. It can be said that organisms with lower reproductive efficiency tend to have longer lifespans. Conversely, animals like mice, which grow quickly and reproduce in large numbers, do not need long lifespans because they can complete the task of transmitting genetic material DNA in a short time. The principle is that individuals who have completed their reproductive tasks have no further purpose and thus have shorter lifespans. This theory is known as the disposable soma theory, and most organisms' lifespans generally follow this principle. The maximum lifespan of mammals is generally about 9 times their age at sexual maturity and 6 times their developmental period. Mice can reproduce their first litter around 3 months after birth, so they typically live less than 3 years. Humans attain reproductive capability at 14 to 15 years old, and growth ceases around 20 years old, making the human lifespan approximately 120 to 130 years. This aligns with the current record for the longest-lived human. Humans are not unique compared to other organisms. This explains why humans have a lifespan limit from a natural evolution perspective. Like most animals, based on finite resources, genes set a mechanism for old individuals to die. However, not all life forms have a lifespan limit. Some ancient life forms, such as certain bacteria, some jellyfish, flatworms, and even some lobsters, do not have a lifespan limit. Yet, too many predators and cooking methods prevent most of them from living long, so their genes have not evolved a lifespan limit. Therefore, aging is likely a genetic setting to limit lifespan, avoiding prolonged survival of old individuals that consume resources and hinder gene propagation. Lifespan limitation, like other biological traits, is a product of natural selection, a method for more effective gene inheritance. While a lifespan limit does exist, knowing this need not be discouraging. Humans have already understood the principles of gene transcription, translation, and selective expression through natural selection. If we understand which genes control human aging and how they do so at the gene expression level, we can truly combat aging and death. The following content primarily comes from David Sinclair's book Lifespan. Sinclair, a top professor at Harvard Medical School, discusses epigenetic disruption and, more specifically, the breakdown of life circuits as the main causes of aging. Previously, I emphasized that our bodies are structured based on genetic information, while our personality and ways of thinking are shaped by the environment. The brain is molded into a specific character by the environment it encounters. This correspondence is still determined by genes. The proteins expressed by genes construct cells, realizing all cellular functions, and humans are made of cells, so genes determine everything about us. However, for multicellular organisms, there is an obvious issue Although each cell in our body contains a complete set of genetic information, our body consists of various types of cells. They all have the same genetic blueprint, but the results are vastly different. When so-called cell differentiation begins, our genes start selective expression. But how is this selective expression specifically chosen? Sinclair uses a vivid analogy. Genes are like a piano, and different cells are like different musical pieces played on it. However, without a pianist, the piano itself makes no sound. So, who is the pianist? It is various substances within the cell encountering different environments, such as cold, heat pathogens, or abundant nutrients. These trigger specific proteins to break free and go to the cell nucleus to combine with specific gene promoters, initiating the expression of those genes. For instance, during the human embryonic stem cell stage, each cell is subjected to different directions of compression, different amniotic fluid concentrations, and various nutrients. This activates different genes, leading to cellular division of labor within the body, creating different musical pieces. But can the pianist hit the wrong notes? Many past scholars believed that aging was caused by the accumulation of DNA replication errors within the body, indicating a problem with genetic information. The first successfully cloned mammal, Dolly the sheep, was frail and sickly, which was explained as premature aging. It was thought that the DNA taken for cloning had already accumulated errors, causing Dolly's premature aging. However, many years later, the cells from Dolly were used to clone another generation of sheep, which were very healthy and lived as long as any normal sheep would. As gene sequencing technology matured, scientists sequenced the genes of many elderly individuals and found no significant changes compared to their younger selves. So, what changes occur at the cellular level as people age? The issue lies with the pianist, who gradually transforms from a graceful maestro into a chaotic music fanatic, playing the body's music erratically. Genes that should be expressed become silent, while those that shouldn't be expressed become active. Ultimately, the body loses its original order, can no longer properly repair environmental damage, and the likelihood of various diseases increases. This is how the wall of death comes at you. This phenomenon is called epigenetic disruption in biology. Epigenetics is the study of gene expression in contrast to genetics, which studies the changes in life after gene sequences are altered. Epigenetics examines the factors within cells that cause changes in gene expression without altering genetic information. Since genes do not change before and after aging, it is epigenetic changes that have caused significant issues with the pianist of our DNA. When life first appeared, ultraviolet rays began to harm the bacteria at the bottom of a drying pond, waiting for the tide to rise. They had to endure this period, but many bacteria's DNA was broken by the ultraviolet rays, urgently needing repair proteins within their bodies to fix the DNA. However, another unstoppable behavior of bacteria was disrupting this process: reproduction. When the proteins responsible for DNA repair started working, helicase and DNA polymerase would also arrive at the damaged site. These proteins would get in each other's way, and reproduction, which usually takes priority, would interrupt the repair process. This resulted in the newly replicated DNA being damaged, leading to improper gene expression in the two daughter cells, ultimately causing the bacteria to die. We often say that survival and reproduction are the basic needs of living organisms, but at this point, these two needs are indeed contradictory. The contradiction caused death, and death allowed for the selection of bacteria that could repair DNA by actively stopping the replication process, allowing them to survive this existential battle. How did they do it? Shutting down the reproduction process also required specific genes to express proteins that block replication. However, this gene could not be constantly expressed, otherwise the bacteria would become extinct. It needed to be expressed only when DNA was damaged and remained silent at other times. This is where evolution showed its magic. The DNA repair proteins evolved to bind with genes that inhibit cell division and reproduction when no damage occurred, preventing transcription by holding these genes down and blocking their expression. When DNA damage occurred, these repair proteins were displaced in a chain reaction, allowing the inhibition of reproduction genes to express. This expression produced proteins that blocked DNA replication, halting reproduction. Once the repair proteins finished their job, they returned to suppress the inhibitory genes, stopping their expression and allowing healthy cells to reproduce again. This is a complete life circuit. Early bacteria used this circuit to resolve the conflict between survival and reproduction. The proteins that can suppress gene expression are the ones manipulating the DNA piano. Over time, this process evolved into multiple versions, precisely regulating the balance between reproduction, growth, and body repair. For example, conflicts between cell immunity and replication, nutrient acquisition, and energy production, and cell growth and function were all resolved through this mechanism. Moreover, the most critical repair work was not limited to DNA but also included many vital cellular components, such as cell membranes and organelles. The repair proteins here even include telomerase, which repairs telomeres. When DNA replicates, it needs a starting sequence, but this sequence is not copied, so the replicated DNA is shorter. The DNA starts and shortens with each replication until there is no starting sequence left, preventing the cell from replicating. These multiple consecutive starting sequences at both ends of the DNA are called telomeres. Telomeres shorten until they disappear, making cell replication impossible. This was initially why scientists believed humans had a lifespan limit. However, a type of repair protein called telomerase is expressed in various stem cells with division tasks. It can repair telomeres like DNA repair, so telomeres are not the ultimate determinant of lifespan. The real issue is that this mechanism is not entirely reliable. Repair proteins fixing DNA might get lost in the cell's complex maze, especially as damage increases. After these repair proteins work hard to complete repairs, they may not return to their proper positions to inhibit gene expression. As such events become more frequent, genes that should be silent continue expressing, while those that should be expressed remain silent. Cells become disordered and can no longer function properly. For example, lacking telomerase ultimately causes stem cells to stop dividing into various cells, marking the onset of cellular aging. This raises another evolutionary logic question. If repair proteins are so crucial, why don't cells produce more of them to replace those that get lost, maintaining normal cellular functions and preventing aging? The issue is that if humans didn't age, cells would produce large amounts of repair proteins until they reach their developmental peak at around 25 years old. However, as we enter middle and old age, the expression of these repair proteins decreases. Accumulated damage increases, but the amount of repair proteins doesn't correspondingly rise and even slightly decreases. Additionally, each cell's DNA contains a specific blank segment that doesn't participate in protein expression but gradually accumulates more methyl groups as the cell ages. The older the cell, the more methyl groups it carries. Scientists now use this methylation level to determine a cell's age. But it's not just scientists. The cell itself uses methylation to change its epigenetic characteristics based on its age. Why does a cell need to know its age? Is it to decide whether to repair itself? When it's time to die, it stops repairing itself, reducing the expression of repair proteins, and becomes more disordered until death. This conserves resources, allowing the next generation to thrive. The book lists several longevity methods that apply to all organisms, proving this hypothesis. The first method is to reduce caloric intake appropriately, known as caloric restriction. Our current sense of being full actually exceeds our body's needs. In prehistoric harsh environments where food was scarce, eating more was beneficial. But in reality, eating balanced meals to about 70% full provides enough nutrients for all bodily functions. Always eating to fullness signals the body that the environment is nutrient-rich, prompting quicker completion of reproductive tasks. Once reproduction is likely completed, there's no need for the individual to live much longer. Repair proteins are no longer abundantly expressed, and various aging-related diseases prepare to take over. Conversely, limiting caloric intake signals the body that food is scarce and offspring survival is low, making it unsuitable for reproduction. This increases the expression of repair proteins, allowing the individual to live longer and reproduce when resources are plentiful. Reducing caloric intake appropriately without causing malnutrition can indeed promote longevity. Sinclair found through experiments that long-term reduction of caloric intake by 30% in mice and monkeys led to significant health improvements, reflected in blood pressure, blood lipids, and fur condition, with a notable increase in lifespan. Among 20 rhesus monkeys subjected to caloric restriction since 1980, 6 lived to 60 years old, whereas their typical lifespan is 40 years, representing a 50% increase in lifespan. Human experiments yielded similar results. Due to human nature and abundant resources, participants managed to reduce caloric intake by about 12% over 2 years. Post-experiment, participants' physical fitness, heart, lung, and blood tests indicated younger states, with some showing increased telomere length in stem cells. Similarly, moderate exercise and cold exposure, which put some pressure on the body, made it appear younger by increasing repair protein expression, ensuring the stability of life circuits. This effect is universally applicable from lower organisms like yeast to fruit flies, nematodes, and mammals like dogs and monkeys, which is extremely rare in biology. Another principle that meets these requirements is called the central dogma, which is the process of DNA transcription and translation into proteins. Sinclair refers to the genes expressing these special repair proteins as longevity genes. Every cell in every person has these longevity genes. The difference lies in whether they are activated. Under the ruthless setting of genes, they choose to remain silent, no longer being played, leading to our aging. The next step is to figure out how to trick these longevity genes into staying active. If this viewpoint is correct, humans would theoretically have no lifespan limit. Once we identify the culprit behind aging, what should we do next? And if we indeed find such a method, how should human society cope with such a monumental change? These topics will be discussed in the next episode. That's all for this episode. If you enjoy our content, please subscribe to our channel for more exciting content.